This video is going to look at the basic structure of DNA. So this is going to include ideas about these words that you can see here. So the twisted ladder structure of DNA, otherwise known as the double helix. And we're also going to look at the individual structure of DNA, including a nucleotide. And also we're going to look at the relationship between DNA, genes, and chromosomes. So just starting off, what we can see here is these black lines around the outside, and you can see that they are they kind of come in a pair, or like two parallel lines that are coming together. These black lines form almost like the outside parts of the ladder, or we also call these the backbone of the ladder, or the backbone of DNA. And in a moment we'll look at what actually makes up that backbone of DNA. And then in between these two backbones are almost like the rungs of the ladder. But you can see that how I've drawn them here, they're actually made up of two parts of the DNA coming together. So it is not one continuous component running the whole way across. It is the paired up structure that forms together. You can see over down here in the bottom left of the page that we've got a close-up of two backbone parts of the DNA, and then we have these rungs coming across, once again made up of two different components here. And what you can see, the difference between the almost two-dimensional type structure of DNA here and what it actually looks like in real life is simply that there is a twist in this molecule. So DNA, when it unravels, as it does from time to time, looks like this, as two backbones with the rungs in between them. But when it reforms into its more natural state, you'll see that there is a twist in it, and this twist forms this double helix, otherwise known as a twisted ladder. So we're going to look at more detail at this idea of the DNA structure now, and looking at what a nucleotide is. Okay, I'm going to talk about that in a second. So this image here is the same as what we looked at over the page. So once again, we've got the backbone, or the outside of the ladder here, and the rungs going across. And what you can see, and what you might notice, is that I have paired up these parts of the rungs in different colours. So in this case, red is always going with blue, and yellow is always going with green. And the whole way down, they will always be in that same kind of relationship. And we'll look at that those pairing rules in a bit more detail shortly. If we're going to look at a little bit more detail, a zoomed up version of one half of the rung and the backbone, this is what we're looking at over here. Okay, so I've labelled these for us already, and we can see that a zoomed up version of here, the blue part here, the blue line part of the rung, corresponds to this. Now this is a base, so I'll bring the label down there a little bit closer, and this base can be one of 4 different types of bases that DNA has. We'll look at those names shortly, but they are adenine, which is represented by A, thymine, represented by T, cytosine, represented by C, or guanine, represented by G. Okay. We also have over here, we have this pentagon shape, and this is a 5-ring sugar, carbon-based sugar. And if I'll just move that name up a bit closer, so this is the sugar part of the DNA, and this is actually where the name for DNA comes from. So this particular sugar is called deoxyribose, and this gives DNA its name. So deoxyribonucleic acid, that's where we get the DNA from. The phosphate over here is the black part on the diagram. Now, one thing I want to make clear is that this black backbone here of the DNA is not just the phosphate. It's actually made up of alternate phosphate sugar, phosphate sugar, phosphate sugar, and we'll see how those connect in a moment. But just to make it very clear, the phosphate and the sugar, so if I draw a line here just to split them up, okay, so this part over here will be the backbone part of the DNA, and it is only the base, in this case an A base, adenine base, that forms the rung coming across into the middle. So this is a sort of enlarged version of what we were looking at previously, and what we can see now is that we have these alternate phosphate sugars that I was just talking about. So phosphates are these circle parts, and the sugar is the deoxyribose sugar. So phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. Okay, the alternate phosphates and sugars are going to make up the backbone of the DNA, or the outside of the ladder, and then poking out into the rungs of the ladder, or forming the bases of the DNA, will be these bases. So I've put all of them down here, but we have adenine, thymine, cytosine, and guanine down here. What you will notice is that I have drawn in a very schematic way that these bases have different shapes to the end of them. Okay, so this is certainly not representative of what these molecules actually look like, but on a schematic point of view, it indicates that each of these bases is different from each other, and this starts tying into this pairing rule. Okay, so at the moment, we're actually only looking at half of our DNA molecule. So we have our backbone here, and we have the bases that become the rungs of the ladder here, but we are missing the other half of the DNA. So at the moment, we just have one strand of DNA, but DNA sort of sits naturally as a double-stranded molecule. When we add on that double strand, and we'll do that in a second, you'll see that there is a very particular way that these bases must pair together. Okay, so we've got them doubled up now, so this is now a double stranded molecule of DNA. Once again, we've got one backbone here, or one outside part of the ladder here, and then we can see that there is another one now on the other side. So the red and the black portions of the diagram represent the outsides of the ladder, and then coming into the center we have these rungs, or these bases, coming in from either side and meeting in the center of the DNA molecule. You can see that now it's starting to make a little bit more sense about how these bases go together. So you can see that adenine and thymine fit very nicely together. Just like down here, we have cytosine and guanine fitting nicely together as well. So this is what we call the base pairing rule, in that adenine always pairs with thymine and cytosine always pairs with guanine. As like extension material for year 10, what you might notice is that the direction of the sugars is actually reversed on each side. So in this direction you can see that the pentagon of sugars is pointing upwards as we go down, and then on the other side of DNA, the pentagon of sugars, the point is actually going down in the other direction. So this is a little bit beyond year 10 level, but this is hinting at the idea that the strands of DNA actually run in opposite directions to each other. But as I said, not necessary to understand at a year 10 level. Okay, so let's just reinforce this base pairing rule. And so I'll write them out in full at the moment. And so we will say that adenine always pairs with thymine, otherwise known as A always pairs with T, and cytosine always pairs with guanine. C always pairs with G. This is always the way that it is in DNA, and as we touched on previously, because of the particular shape of these molecules, they can never pair the other way around. So if you see an A on one side, there will always be a T corresponding to that, and likewise with C and G, they always get paired up together. So the last thing I wanted to do is just touch on this relationship between DNA, genes, and chromosomes, because this is one of the hardest things to kind of get your head around. So over on the side of the page here, we've got the image that we were looking at the very beginning, which is this double helix shaped molecule of DNA, this twisted ladder of DNA. Once again, the black lines represent the backbone of the ladder, or the phosphate and the sugars running down the side, and in the center we have our rungs coming across, which, as you know now, will be the bases of the nucleotides matching up in the center. So this is DNA. So DNA is a chemical molecule that contains coding information for our traits and characteristics. If we are to look at what a gene is, a gene might be it's a section of DNA that codes for a particular trait. And as slight extension material for year 10 also, it is actually the sequence of bases, so the order that these A, T, C, and Gs are in, the specific sequence that is going to determine the specific gene that it happens to be. But let's just write down that this is a gene here, and this here would be our length of DNA itself. Okay, so that's not too bad to get your head around, but the next part is, how does this relate to the chromosome structure that you can see up here? So the easiest way to think about this is that the chromosome is simply a highly packed form of DNA. So the chromosome is DNA, but it is just a highly coiled up structure. So what you can see here in this part of the page, and I'll just zoom into that for us for a little bit, what you can see is that we've got a diagram here that looks very similar. So we've got these base pairs matching up to, across each other, and we've got the backbones running down the side. This coils up into our DNA, so it's got this natural twist to it. Now DNA, if it was like this state in our cells all the time, it would get awfully messy, and there would be a huge amount of DNA just hanging around in our nucleus of every single cell, and so there needs to be a way to package it up so that it takes up less space. It becomes a more discrete unit. Okay, so DNA here, what happens is that it actually gets coiled up upon itself with the help of some little proteins here, and these are called histones. You don't need to know about the histone proteins, but basically it gets coiled up upon itself, and then these coils get coiled up upon themselves, and then they get coiled up upon themselves. And so you can see in this image up here that the parts of the chromosome that we can see under microscope are simply highly coiled up parts of DNA. Okay, so let's just bring that down so we can just sort of point that out to us. So the whole length of the DNA, as you can see here, will be coiled up. So the chromosome is simply coiled up DNA. So you could say that sections of DNA, which are the genes, are also found in chromosomes. So if I just zoom into here one more time, I could actually draw a little band across the chromosome here, and that would represent a gene. So this is a slightly confusing part, because genes can be represented, like we can see down here, as a section of DNA, but genes are also a section of the chromosome, because a chromosome is simply a coiled up bit of DNA. Okay, so hopefully that has clarified a few ideas. So just running through those definitions again, you might like to write these down. DNA is a chemical molecule that contains genetic information that codes for traits and characteristics in our bodies. A gene is a section of DNA that codes for one particular trait or characteristic, and the chromosome is a coiled up version of the DNA, but it is still DNA in itself.